morning. I'm Dr. Chattopadhyay, Senior Vice President with IKP, and I have the privilege to welcome all of you to Hyderabad and to IKMC. It's a wonderful morning. It's a wonderful early wintry nip in the air kind of morning at Hyderabad. And what better way to start a Saturday morning than to have a host of dignitaries coming in from multiple spaces across the innovation ecosystem, the academia, the investors, the innovators, all of them with a singular goal to exchange thoughts and to come together and start looking at how we can gain insights into how the innovation ecosystem can move forward. I now have the privilege to invite our CEO and chairman, Deepanwita ma'am. Ma'am, would you take the dignitaries and the participants on to what the journey of IKMC and IKP has been? Over to ma'am.
Thank you, Ambuj. A very warm welcome to all of you who are gathered here today to celebrate innovation. When I say celebrate innovation, I really mean it, because we today have someone with us, and I mean Dr. R.A. Mashelkar. He's been one of the founding directors of IKP Knowledge Park. Thank you, sir. Thank you for being with us. And it's such a pleasure to have Dr. Mashelkar with us. I'm not going to take time by thanking and naming each one of you, saying thank you for being with us, because as the program rolls out, you'll meet each other, you'll know each other, and that is the idea. I was going through some of the statistics of our 22-year-old journey of IKP. And what strikes me is 2009, in October 2009, we had 41 startups and mid-sized large companies that we had supported at IKP. 2019, October, it was 650. And today, as I stand in front of you, you won't believe it's 1,300. So what makes it like this? There's this exponential increase. And I was asking myself, is it because of IKP, the internal team? Is it because the ecosystem has changed? Is it because now we are not alone? When we started in 1999, we were the first science park in India, life sciences park in India. There were very few incubators, and now I can tell you the number of incubators in India has crossed 700. Science parks are not so many, but that also is getting set up in all the major engineering institutes and universities. It could not have happened. This kind of increase in the number of startups that we have supported, and if we have supported 1,300, it would be at least 10 times more that entire India is supporting. And that tells me that it could not have happened without the participation and collaboration of a large number of academic institutes, innovators, and startups, mid-sized companies, large companies, and enablers like us. It gives me great pleasure in saying that what we have done is fantastic. What we need to do is much more thought-provoking. And what is that? What is the next? So the question today and tomorrow we are going to deliberate on is, what is the next? What do we want the country to do in terms of innovation? What do we want players like IKP to do? And there are several incubator CEOs present here and other enablers present here, what is their role? How can we help the ecosystem develop further? The way we have structured the conference is to ask ourselves this question again and again and again in different forms, so that at the end of the 2 days, we go back with an action plan. We had a strategy meeting 3-4 months back, and this is kind of, and Utkarsh is here, Zohar is here, and I see some of the people who are present in our strategy meeting. This is the first action play of the strategy meeting, so to say. We are actually saying, this is what we have done, and this is what we are going to do. I want to end my talk by saying that out of the 4 very important founding directors of IKP Knowledge Park, Mr. Narayanan Vagul, then chairman of ICICI Bank, was supposed to be here with us, but he could not make it because of ill health. Dr. Bala Manian, who is joining us online from US. You'll hear Bala, and many of you know Bala, the amount of energy that Bala exudes. While we are missing that, Bala is with us all the time. Dr. R.A. Mashelkar, Dr. Mashelkar is here with us, and Dr. Ashok Ganguly. Dr. Ashok Ganguly could not travel, but sir, I don't know whether you've seen his memoirs that came out very recently. It's called Afterness. And in the memoirs that I read, he had an illustrious career, but there is half a page where he actually has mentioned Knowledge Park. And it gave me such pleasure that in all the kinds of things Dr. Ashok Ganguly has done, he felt that IKP made a difference. And if that is the commitment we have of our founding directors, I think we need to carry that forward. And with that note of Dr. Ashok Ganguly, I think it is time to hear the keynote addresses and first welcome our chief guest, Dr. R.A. Mashelkar. So, with the keynote address, would you like to sit there to see the presentation? Because this is small. Yes. So can I invite Vikram? And the reason I'm inviting Vikram, Dr. Vikram Gopal, you'll hear him in other sessions, is to introduce our first keynote speaker, Dr. A.J. Velthuis. Vikram?
Thanks, Deepanwita. Can you guys hear me okay? Good morning to everybody. Well, I get the pleasure to introduce our first speaker, A.J. Velthuis. I can go on with his accomplishments and everything else, but I think the way I remember him is we were in the midst, during the midst of pandemic, you know, just from my introduction standpoint, I'm the Chief Technology Officer for a company in the US called Ascent Performance Materials. We are the world's largest integrated polyamide suppliers, okay? And during the pandemic, we had actually, or during the last 6 or 7 years, we had actually developed an antimicrobial polymer. And we, as the pandemic came along, of course, the big thing was what can you do with it and can you deploy it the right way? And I was looking out for experts, okay, 2.5 years ago, and I reached out to A.J. Velthuis, this gentleman here, and he was at the University of Cambridge at that time, right? And more often than not, people would say, hey, I don't know you, I don't know what you guys want, and that's where the relationship started, okay? And I've actually had the distinct honor and privilege to knowing A.J. because we put in across the pond from the US to UK, and through him actually testing the material at Mount Sinai Hospital in New York. We are the first ones to do it. I mean, just a tremendous person. He's a virologist by training, one of the best I have seen, very much fundamentally driven. And really, I don't want to take any more time, but I'm going to ask him to come over today. AJ is a professor of virology at the Princeton University in the US. Okay, and I let him come and give the talk as far as the whole, come along, AJ. I don't want to steal your thunder around here, and let you start off.
Thank you so much, Vikram, for this kind and wonderful introduction. And thank you also to the organizers for inviting me to speak here today and tell a little bit about the work that we have been doing in my lab and in the labs of collaborators to better understand RNA viruses, and in particular viruses that are responsible for pandemics in humans. Specifically, we've been looking at 4 broad points, how we can broadly inactivate viruses, and this is related to the collaboration with Vikram, if we can limit their spread, but also, can we understand more about what they do once they get into our body, once they get into our cells, and what they do to cause disease, and what then subsequently happens to these viruses as they adapt to us and mutate. Now, let me kick off with introducing these various viruses that I'll be talking about. You may have heard of SARS-CoV-2, the agent that is causing the current pandemic that we're in. And like flu, it has this sort of more circular, spherical structure with key proteins on the outside that are important to attaching to our cells and getting into our cells to allow the virus to replicate it. It is then surrounded by a membrane that protects the inside, and the inside is quite important, and I'll be talking about those contents quite frequently throughout my talk, because that is the genetic information of the virus. It allows the virus to express proteins, copy itself, make new virus particles, and then jump to the next cell, jump to the next host. But like I said, viruses come in many different shapes and sizes. I've also put up in the middle here a picture of an Ebola virus. As you can see, this is no longer spherical, it's more elongated. And as such, it needs different mechanisms to get into our cells, because this big floppy thing just is much harder, has a much harder time to get into our cells. And then as a third example here on the right, I've put up a picture of a rotavirus. And what this virus does, it has many layers of protein, almost like an onion, to help protect those contents on the inside. And you imagine sort of what this virus needs to go through. It infects us through the oral route, and then needs to pass through our stomach to infect our intestines. And so it has to survive this really harsh environment, and that's why it has created this capsid of many layers of proteins to remain stable. And so knowing all this information about these viruses helps us to design better strategies to fight them and inactivate them. But as they are different in shape and size and structure, they also cause different kinds of disease. On the right-hand side, I put up some images of an infected lung tissue, and this is what you typically see in an influenza virus or a SARS-CoV-2 infection. Our lungs are alveoli, are thin layers of cells. We have blood vesicles going through them, and big air pockets in between those cells that allow the gas exchange, allows us to take up oxygen and get rid of carbon dioxide. Now, when these cells get infected, they start an antiviral response that attracts white blood cells. And these white blood cells have to go somewhere. And where they go, unfortunately, is in that space where air normally sits. So here in white, you've got the airspace, and in the bottom picture, you see that this has been infiltrated by those white blood cells. And this is part of the mechanism that underlies viral pneumonia and why you get these fluids into your lungs that prevent you from breathing. Other types of disease may present themselves as hemorrhagic fevers or hemorrhagic bleeding, either from skin or in internal organs. and examples, dangerous examples, are Lassa virus and Ebola virus. Other types of disease may manifest themselves in the central nervous system. This can range from rabies virus, which actually changes host behavior, to Zika virus infections that change development. And you may end up with babies that have deformed heads. Now, it's also important to know where these viruses come from and how they may get into our bodies. A lot of work has been done on coronaviruses and influenza viruses, and I'll use influenza here as an example. And what we've learned over the years is that breeding grounds in Siberia in particular, where lots of flocks of birds come together, different species, these grounds are a perfect sort of mixing spot for different bird flu strains to come together and exchange genetic material. Then as winter comes and these birds start to migrate, for instance, to Western Europe, in a country where I am from originally, the Netherlands, sees a lot of these birds going to the wetlands. And what then happens is they drop their feces and spread this bird flu to chickens or poultry that we may be growing and harboring as livestock. So understanding where they originate will help us then figure out how these viruses mutate and mix. But of course, we also bring animals and the virus that they contain to ourselves. Examples are here the wet markets in China, where many, many different species, not only from China, but also from elsewhere in the globe, are brought together and quite frequently slaughtered on site, allowing various fluids to mix and carry this virus from animals to humans. But also, somewhat less harmful maybe, first sight, are country fairs in Europe and in the US, where, for instance, pig raising is done. But these pigs are in pens and they all interact with the fences and then are able to shed virus via those enclosures and spread this virus to other pigs, but also to humans. Now, in addition, I mentioned it is important to know how the viruses exchange genetic information, because this gives rise to new species that we've never seen before, and so our body doesn't know how to respond to them. Again, flu is a good example to illustrate this. The flu genome is broken up in pieces, we call segments, and if a virus, like this red one, infects the same cell as the virus that has this blue genome, the offspring viruses may have a mixture of blue and red genes, and this gives these viruses new features. It may allow antiviral resistance to spread more easily through the population. And more importantly, it creates new genetic makeups that our body just hasn't seen before, and so has a bigger problem to deal with. And just to illustrate how important this is, you may remember the 2009 influenza pandemic. And what we've learned from looking at genetic information is that this virus that came from swine and then started to infect humans was actually the 1918 pandemic virus that had gone through many generations of adaptation in humans, then disappeared into swine in sort of the 1990s, mixed with the bird flu strain, and created a new genetic makeup that then jumped over to humans and started to cause a new pandemic. So knowing that history, knowing what they can do and what the principles are behind this genetic mixing is important for what kind of virus we may see in the future. Now, as I mentioned, these different fundamental properties of viruses, we can target those and inactivate those with very specific approaches. For instance, antivirals or antibodies or vaccines. But there are also more general approaches, and we've been working together with Vikram to better understand how they work and how we can utilize them to inactivate viruses. And one relatively well understood approach is UV light, particularly UVC, which has a wavelength of 254 nanometers right here. And this has the ability to create crosslinks in the viral genetic information. So you can see here there are two building blocks of viral genetic material, and upon exposure with UVC, they are linked together. And what this causes is that when the virus tries to copy its genome, it encounters these structures as roadblocks. You can see that quite nicely here in this graph, where you can change the amount of UVC exposure over time. And all these dark spots are basically stalled acts of viral replication. So the virus runs into these roadblocks and doesn't continue copying the viral genome, and eventually it just fails to spread. So this is a very generic way to inactivate them, and this works regardless of what kind of shape or structure these viruses are in. And there are many different tools and techniques that have incorporated this ability, and I encourage you to go to discussions by my colleagues Vikram and Chris later today, where they go more into detail about how they work and how you may employ them now and in the future. An additional technique that Vikram also alluded to is using antimicrobial compounds into fabrics. Specifically, what we worked on with him was how zinc can be incorporated into nylon fibers. And zinc, copper, divalent methods like this have an inactivating property, but we don't really understand why. So what we did is we developed a testing protocol to see how viruses can be inactivated within woven materials, within fabrics. The problem is that when a virus gets in, it gets absorbed to the fibers, and it's quite hard to get out. So we had to optimize a buffer, a washing medium, if you like, to tease those viruses out without actually inactivating them in the process. And we were able to do that, and then ask the question, is there actually inactivation as they touch the fibers? And then what is the process behind it? And we find that a nylon without zinc doesn't inactivate the virus, but if you add zinc, done by Asst in the US, you get about a 2-log drop in active virus, so virus that is still able to infect cells. We then looked at this virus, we extracted it from the fabrics and asked them, what is wrong with it? What has the zinc done? And what we find is that those proteins that I introduced at the beginning, that sit on the outside of the virus and that are important for getting into our cells, they are destabilized. We don't know exactly why they are being destabilized. We're still investigating this using various microscopy techniques, but you can see here on the right bottom that the amount of intact protein has been reduced, and so the virus has less of this protein available to bind to our cells. And that is just a very easy, simple mechanism through which you can stop it from spreading. Now, we also want to understand disease, and to do that, we compare different types of viruses. We have, particularly for influenza, we have different seasonal strains that still cause a lot of problems, but are more or less harmless to most of us. If you contrast that to pandemic and avian influenza viruses, they have a very high mortality rate, sometimes up to 50%, and they have known, in particularly the 1980 pandemic virus, to kill a lot of people. So I'm standing on the shoulders of giants, so a lot of research has gone into trying to understand what is so special about these pandemic viruses, and what we've seen is that a lot of factors contribute to this. Underlying disease, age, co-infections, but we also know that the immune response plays an important role, and it's partly because you get this infiltration into the lower lungs and it causes viral pneumonia. But what is really the trigger? That's what we wanted to understand. Why are pandemic viruses different in that sense? So the immune response, normally it's there to prevent the viral infection, and if it works well, it's great. If it's too weak, of course, the virus takes over, and if it's too strong, you run into problems like pneumonia. So what we did is look what's happening with the viral genetic information. We know that this is what our bodies sense when you have a viral infection. And it's in a way that doesn't make sense, because the viral genetic information, as I draw there, doesn't exist. Rather, the virus wraps it up into a nice package to keep it protected, so our cells can't really see it. And what we found is that there is a different form of this RNA that is specific for pandemic viruses that exposes the RNA to our cells. And I'll show you quickly how we got to there. So we took different viruses, and I particularly want to highlight here the 1980 pandemic virus and an H5N1 avian virus. What you see at the top is the normal genetic material that is being amplified. But what you hopefully also see is that lower down this gel, in which we like separate bits of genome by size, they're smaller products that are really, really a lot smaller, about 50 to 80 nucleotides in length. And if we then compare how pandemic viruses and avian viruses are different to human adapted viruses, we see that there are a couple of adaptive mutations, particularly in the complex that copies the viral genetic material. And when we put those adaptive mutations in seasonal viruses, we see that the ability to make these additional products, these smaller bits of RNA, you could call them garbage if you like, they trigger a very strong immune response, and they are specific for pandemic viruses like the 1918 virus, and if you put those adaptive mutations in that virus, it loses the ability to make these products. Now, why is that the case? Well, I'll come back to that at the end. But we observed a couple of other key things. Not all those products, not all that garbage is important. So we need a couple that are produced. So from this whole cloud, we can now, based on primary sequence, assign which ones are important for the immune response. And then say, oh, these are the ones we need to look for, see if they're present, and that will tell us something about what kind of disease someone is going to experience. And we figured out the mechanism of why they are so dangerous. And what we saw is that if you have one that's like a small RNA that is able to trigger an immune response, the replication complex stalls on it, very similarly like on UVC. Whereas on a normal shorter RNA that has none of this particular sequence recognition signal, it just carries on to the end. And this links back to that exposure of the RNA to our host cell, with you have a normal replication complex that makes an error, it creates a shorter product. The virus still tries to copy itself because it cannot really distinguish between normal RNA and this garbage stuff. It gets stuck on it, and then you have the structure sitting at the end that is the perfect recognition signal for our host cells, and then they start to trigger the immune response. And we can make viruses that have the ability to make these RNAs, like wild-type H5N1, and that virus kills mice very quickly, but we can also put a point mutation in that virus to reduce the amount of this garbage that it produces. And as you can see, most of the mice then start to survive. And we also see that the immune response is not triggered as much. So there is a clear correlation between how well the replication complex works, if it's efficient, if it doesn't create garbage, and how much our cells are able to see what the virus is doing and respond to that and thus cause disease. And interestingly, a recent paper was published showing that this principle also applies to coronaviruses, and particularly in the SARS-CoV-2 infection, where this virus also makes short bits of genetic material, garbage RNA if you like, and that this is also able to induce a very potent immune response, and that may explain why you get this massive influx of white blood cells into our lungs. Now, I mentioned that we want to detect these RNAs in patient samples and then create the ability to say something about disease progression. Normal techniques like RT-PCR, next-generation sequencing, are not ideal for this, but because we've now identified exactly which species are important, we've started using a technology that is based on Cas13. You may have heard about CRISPR, Cas9. This is a way to edit DNA, but there is also an RNA version called Cas13, so this can recognize specific RNA sequences. And in addition, this enzyme depicted over here can bind the target RNA happen in the second step, and then cleave it, but at the same time also cleave a fluorescent probe that we've added to the mix. So the more cleavage you observe, the more Cas13 recognizes the target it's looking for, the more fluorescent signal, and so you can measure exactly how much of an RNA species is present. And just to cut to the chase, we can detect this garbage RNA even if in 0.2 nanograms of RNA. So it's quite powerful when we don't need amplification steps. So we hope that this may develop into a useful screening method for figuring out which viruses are dangerous for us and what is happening in patients in the future. So I'll try to generalize some of the principles I've talked about now. So we have viruses that are adapted to a certain environment, like a bird. They're happy there, but then they're brought into contact with humans, and this creates the opportunity for them to jump over to us, spread, or a zoonotic event, spillover. But it encounters an entirely different environment. We have ways to prevent this spread, of course, but that requires some changes in our behavior, whether we have these wet markets, etc. Now, this different environment, for instance, difference in temperature, difference in cells, makes the virus behave in a way that leads to a lot of garbage. This causes disease, could lead to death, but it's not sufficiently inhibitory for the virus, so it will still spread to the next human and from human to human. But we have our immune system that starts to generate antibodies, so it starts to put pressure on the virus. It limits spread. The virus will then need to optimize this, so it will start to evade those antibodies, which is exactly what we're seeing with the current pandemic. We have to come up with new vaccines every time. And these mutations typically appear on those proteins on the outside of the virus that we can, for instance, target with zinc. But there is an upper limit to what it can mutate. At some point, it has mutated so far that those proteins are not able to bind to our cells anymore. And interestingly, flu, particularly the H3N2 strain that has been around with us since 1968, has got to that point. It is not efficiently binding our cells anymore, but it's still spreading. So how does it do that? Well, one way is to mutate the replication complex. Make sure that that works so efficiently that it's pushing out enough virus particles so some will make it to the next host. And that's why we see that also garbage starts to disappear and these viruses become less virulent. And so with that, I would like to thank you for your attention. Of course, thank the members of my group who have contributed to this work, our wonderful collaborators, particularly Vikram, and funding that we've received from Wellcome Trust in the UK and NIH in the US. Thank you for listening.
Hello, thank you very much for that wonderful talk. It's a great overview. I would have three questions that came to my mind as you were talking. Number one, you talked about the sensitivity of the temperature and the environment. How is the climate change that is going to affect the basic characteristics of the virus that you talked about?
That's a really wonderful question. Thank you for asking that. I think it has a number of implications. One, viruses have to survive in the environment. So they need to either transmit to air or spread via fomites on surfaces. So their ability to survive on this surface, for instance, longer at a higher temperature, they need to optimize for that. So they need to become more stable. But we also see that, for instance, viruses that spread through mosquitoes, they are also changing their stability because those mosquitoes also have to move to different temperatures, different areas. So spread in that sense is also changing as function of climate change, unfortunately.
Yeah. So the second question that I have is, we talked about pathogenic and non-pathogenic viruses. If we think in terms of our recent knowledge expansion with regard to EBV, Epstein-Barr virus, we used to think of them as being not greatly pathogenic. And recent research is now pointing out that EBV is somewhat responsible, and there's a very strong correlation to the onset of multiple sclerosis associated with EBV. Are there things that we need to be worried about in terms of the interaction between the immune system and the viruses? What we consider today as not pathogenic may eventually, at some time in the future, may give rise to immune response that may lead to undesired characteristics.
Thank you. That's again a wonderful question. I absolutely agree. I think we just don't know enough about what these viruses are doing with us. We think of respiratory viruses as viruses that mostly affect our lungs, but they can spread beyond the lungs. They can go to the central nervous system. And particularly now with SARS-CoV-2, we see effects on the brain, particularly in terms of long-term COVID. And with other viruses, they may have, similar effects that we just have never appreciated. And I think there is a lot of work for us to do still to try and untangle certain diseases that we see and maybe link them to the effects of viral infections.
The last question that I have before I ask the audience to say if any of them have any questions is that with regard to the viruses that you talked about, are there conserved regions in these viruses in order to, today, for example, the flu shots, we anticipate what the version of the virus is going to be, and on a speculative basis were developed. Do you think any time in the future we could end up understanding the non-changing portions of the virus and strategies that doesn't require changes as we normally do with other types of virus?
So I think you're referring here to a universal vaccine, for instance, that can target all kinds of flu strains or all coronaviruses. I think we've made, my colleagues who particularly work on vaccines have made a tremendous effort there in that sense. And there is some form of universal influenza vaccine on the horizon, so I hope that this will work. But I also appreciate that viruses, even in regions that they haven't forced to mutate so far, they may end up mutating them at some point in the future if we start to put pressure on them. And their vast ability to explore mutations at a massive scale, I think still forces us to be somewhat careful in how successful those universal strategies are going to be.
On the SARS-CoV-2, for example, we have focused on the mutations associated with the S protein.
Mm-hmm.
Are there mutations also taking place on the envelope protein and the nucleoproteins?
Yeah.
And can the strategies directed towards the other proteins actually they provide some kind of a broad spectrum vaccine?
It's a good question. There are indeed mutations occurring on the nucleoprotein, on the replicase. I think with vaccines, we predominantly try to target bits of the virus that are exposed to our immune system, so that are on the outside. And so we're more limited in what kind of proteins we can show to the immune system to train it. But work is being done, again, I can give influenza as an example because there's so much work into this, that to not only have vaccines targeting excuse me, the hemagglutinin protein, but also the neuraminidase, which is also on the surface. So have a bivalent approach. So your idea is indeed something that is being pursued.
Thank you very much. And I don't, maybe 1 minute, anybody want to ask a question from the audience?